So hello world and hello everyone already who signed on here this Thursday morning, evening, wherever you are in the world. This is our second webinar with Christine Houghton, and she's gonna dig more into the clinical side of sulforaphane and the use layer. I'm Anne-Catherine, co-founder of Nordic Laboratories and DNA Life, a group of companies that wants to change healthcare through functional medicine and personalized medicine, nutrition, lifestyle changes, and so on. So remember, you have your profile in the system where you can get lab tests and you can order supplements. And of course, we will send out a mailing with the recordings, and Christine has prepared the slides, so they will go out hopefully tomorrow as well. And there will be a special offer for you in that email as well. So look out for that one. So last time, for those of you who who partook, Christine elegantly took us through how whole food are superior to just single dosing of antioxidant vitamins. And today she's going to dig into the more clinical conditions. And you will probably hear genes like CYP1A1, quinone reductase, vitamin D receptors, GTS1, GSTP1, and so on. And remember, if you want to know how your genes are and your inheritance in regards to some of the things that Christine is talking about, or you want to find out about it in your patients, there is a DNA health test because it's looking at those genes. So very exciting. So anyway, the star of today's show is Christine Houghton, and she is in Brisbane, Australia, and you can certainly hear that on the charming accent. So she holds a degree in Nutrigenomics, biochemistry, and human nutrition. And she founded Cellular, an evidence-focused company with research and manufacturing nutrigenomically active ingredients. So that's where the sulforaphane is very exciting. Her current interest is in the investigation of phytochemicals which impact gene expression within the human cells in relation to gut ecology, immune modulation, and cellular defenses. And then sulforaphane, as I said last time, is her baby, and she completely agreed because she has published several articles on sulforaphane and book chapters as well. So remember, we have the chat on the side, ask questions when they pop up, and I'll take a note of it. And at the end, I'll do a Q&A with Christine and take your questions up. So anyway, Christine, the stage is yours. Please enter it, and we are so excited to follow you.
Thank you so much, Anne-Catherine, and it's a pleasure to be back again with everyone who's on the other end of my computer screen. So here we are in session 2, just a very brief run through what we did in session 1. We looked a lot at antioxidants, and we looked at different types of antioxidants, and we looked at the fact that really some of what we've been doing for 50 or 60 years is really old science by today's standard. And so now we're looking more at signaling in terms of being able to enhance the cellular defense mechanisms. We talked briefly about exercise, and I'm going to talk a little bit more about that today, and what exactly is sulforaphane. So what are we going to do today? I want to look initially, before we get into the clinical material, is what it takes to get a sulforaphane yielding supplement that can actually do something clinically. We need to look at terms like bioavailability and dose response, and then we'll compare that with the clinical trial data, because the world of sulforaphane supplements labels is quite confusing, and there are a lot of inactive supplements available on the market, and they're not necessarily cheap, so patients can be choosing something and it doesn't work, because it isn't delivering any sulforaphane. Some of the clinical applications I want to look at is detoxification mechanisms in general, and in particular estrogen metabolism. I want to briefly skim over gut health and Helicobacter pylori infection. We could spend all day on that, but we don't have all day. And a little bit on immune defenses, and then briefly on autism. So a little bit of an update from last week. So essentially what we're trying to do when we eat a diet rich in plant food, if we eat an adequate quantity, and to me, we need about 600 grams daily of non-starchy vegetables to give our NRF2, or give our cells the NRF2 activation that they really need. All of the bioactive molecules in plants send signals to the cell. They switch on NRF2, and it's NRF2 which then produces or activates a whole host of protective genes related to cellular defense. At our company in Brisbane, CellLogic, we work on the principles of cellular defense, and if you've seen our logo, which is maybe floating around somewhere on an introductory slide, it looks like this. And each of these hexagons is meaningful in terms of cellular defense. So the first principle we look at is redox balance, and we saw that last week. The other principle, which is really important, is inflammation, which is a branch of our immune function, and both redox balance and inflammation feed into each other in a reciprocal arrangement. Next thing a cell needs to function normally is the ability to produce energy, and then we need to look at detoxification as well. Now, all of those interactive pathways there are all essential for cellular defense. There's one more, and it's methylation. And we set methylation aside because not only is it involved in day-to-day biochemical reactions, but it's also important in epigenetic processes. I want to just reiterate the fact that there's a branch of nutrition science floating around which takes methylation out of its context and treats it as if it is the most important function within cells. It's not. It's just one of a whole host of different processes, none of which can exist on its own, and every one of those must be functioning optimally. And that's my approach to looking at cellular defense. To me, I don't talk about antioxidants because it's embedded in redox balance, and you'll see, or I think we saw last time, that detoxification enzymes and antioxidant enzymes are all simultaneously activated when we induce gene expression. So the thing about exercise is it's a really good opportunity to see how cells respond to a stressor. So if we were going to exercise as this fellow's doing here, we're very quickly going to inhale more oxygen, and part of that oxygen tends to move sideways off to superoxide radical, and that is just about 2 or 4% of the oxygen we breathe converts to superoxide. That is highly pro-oxidant, and it's a stressor, and that sends that signal that we need to the cell to tell it to activate NRF2. So when we do that, as we saw last week, we're going to upregulate a whole host of genes. And if you look at the diversity of these genes, we've got genes coding for glutathione, phase 2 detox enzymes, metallothionein, which is nature's heavy metal chelator, ferritin, which binds up free iron, which we can't have floating around, and the BRCA1 and BRCA2 genes, and hundreds of other cell protective compounds. So that's why exercise is so beneficial. So this study from 2009 really helps to illustrate why the antioxidant compounds or enzymes produced by the cell itself have a completely different effect from antioxidant vitamins or other exogenous sources. So in this example here, 20 young healthy men were given doses of 1,000 milligrams of vitamin C a day and 400 units of vitamin E a day, every day for 4 weeks, and at the same time were doing an intensive exercise program. The researchers were looking at metabolic markers to determine what the effect was. So their hypothesis was, if we got these young healthy men exercising and taking the supplements, would we get an enhanced effect in the group that took supplements compared with those that took no supplements and did exercise only? What might be surprising was that it was only the group doing the exercise that had any beneficial changes in the metabolic markers. And those taking the antioxidant vitamins got no benefit whatsoever from the exercise. And so that's just over 10 years ago, a decade. That really changed the way we seriously thought about antioxidant vitamins. Completely turns on our head what we'd thought before. So just to reiterate, so the cell is constantly monitoring its environment and it's picking up these stress signals. It now sends NRF2 from its binding site in the cytoplasm into the nucleus, and that's where now it activates all these protective genes. Interestingly, the categories we saw before in the hexagons, the redox, the anti-inflammatory, the detox and the energy synthesis are all part of that pattern. And it turns out that the sulforaphane derived from broccoli sprouts is the most potent known phytochemical activator of NRF2. Now, there are drugs which are more potent. One of them, bardoxolone methyl, a few years ago was involved in a clinical trial. That trial had to be stopped because the patients were actually dying so many months into the trial. Why would that be? Well, as it turns out, the bardoxolone methyl is 830 times more potent in its NRF2 activating capacity than sulforaphane. So, I mean, that is well outside the scope of anything nature feels comfortable with. And as it turned out, the patients were dying of heart failure. And an earlier study actually showed that the drug also produced significant loss of magnesium. So I think that's an interesting correlation. So NRF2 target genes are closely involved now in drug research. Pharmaceutical companies are onto the concept, and this diagram here, which comes from a really lovely paper by Guarini. If you're really interested in looking further, this paper from 2013 summarizes a lot of the principles that we're talking about. So part of what it's saying is that nature has grouped the relevant genes into convenient places so that if we activate NRF2 here, we can switch on all of the glutathione producing and regenerating enzymes at the same time. And we'll come back to that again in a minute. These are the NADPH producing. So this produces reducing agents. So this is part of the antioxidant process within the cell. All these genes are grouped together. They're all activated by NRF2. Quinone reductase, which we'll go into in some detail when we get to the estrogen metabolism. It's a fascinating story, this one, and not enough is really being said about it. And then the GST, the glutathione transferase genes, and glutathione peroxidase. These are all genes which require, or their enzymes do require, glutathione itself as part of their activity. And those of you reading Nutrigenomic reports will recognise a lot of these genes as ones which are on your report. Whoops, I just need to go back one. Over here, when we're talking about iron sequestration, you'll probably have ferritin on your Nutrigenomic profile, and you've probably got hemoxygenase 1 on there as well. They're also important parts of the cell's defense mechanism. So these are really key points worth keeping in mind. There's a glutathione molecule, pretty simple really. It's just made from 3 amino acids, glutamic acid, cysteine and glycine. And interestingly, this is a sulfur-containing molecule. And I really want to point this out because I hear a lot of clinicians saying, my patient can't tolerate sulfur, therefore he's got to cut out all his sulfur-containing foods. You don't want to cut sulfur out. You want to figure out how to make sulfur-containing foods be metabolized better, and we can talk about that later if you like. But essentially, if you don't have enough sulfur coming into your diet, you don't have enough sulfur for producing important molecules like glutathione and others. So keep that in mind. There's a whole host of enzymes which are the NRF2 target genes we looked at before. But what's so important about glutathione in this context is that it's most abundant in the cell in its reduced form, and that's the glutathione form, which is antioxidant in its activity. When it does its job, it converts now to the oxidized form, which is pretty useless now, but then it can reconvert back to glutathione. How does it do that? Well, it uses GPX, glutathione peroxidase, over here on this list, and it uses glutathione reductase down here. So all of this is just so beautifully and intricately arranged by nature that it all works in unison. So when I see people wanting to use a supplement of glutathione, I don't understand why they would do that, because what you're really better to do, in my opinion, is to let nature do the work. Activate all of these genes simultaneously, exactly as nature would do. They're all NRF2 target genes, and when you activate NRF2, you switch them all on simultaneously. Let's talk a little bit about dose. This is just a paper that's free online, which I published in 2016. And if you wanted a summary of some of this information, this paper will help you find it. So we're talking about sulforaphane compared with other NRF2 activators, and can the clinician's expectation be matched by the reality? This is about dose, as you'll see shortly. If we are talking nutrigenomically, we need to remember that when we're giving any sort of a supplement with a bioactive content, we need to be able to reach a certain dose threshold for the gene to be switched on. If it's below the threshold, nothing happens. And here we've got the signals coming in from oxidative stress and whatever other environmental signals are talking to the NRF2 in the cytoplasm, so it's got enough activity to allow it to translocate to the nucleus to produce all these protective compounds. I like to look at the analogy of a little child. So here he is, he's desperately reaching up to try and turn on the switch, but he's not tall enough and nothing happens. Whereas over here, he's cleverly increased his height temporarily and now he can turn on the switch. So the dose response is really very important. And I say this and I reinforce it because I see so many supplements on the market which I would say have ingredients listed convincingly on the label that only have doses which to me are just like a wave over the top so they could write it on the label. If there's not enough to activate gene expression, nothing happens. Here's a typical dose response curve. So on the x-axis, we've got whatever the drug or the supplement is, we're going from a low dose to a high. And on the y-axis, we're going to have a look at the effect of that drug. So here's the threshold level here. Any dose above that line, we'll get some action, and any dose below it, nothing happens at all. Up here, we then get a situation where we can reach our peak, but then we're megadosing for no logical reason. So all of this slope on the curve here is the therapeutic range. The next thing we need to talk about is the bioavailability. And this is equally important. And there are lots of barriers to bioavailability, and lots of the molecules that we try to use as supplements we assume are getting into the cells because we swallow them, but that isn't necessarily the case. So when we consume a supplement, then we have the intestinal epithelium. So these are supposed to be epithelial cells. We've now got some intracellular fluid, we've got a capillary, and now we've got the tissue target cells. So for our molecule to do something clinically, we need to get enough of it into these target cells here that we can increase gene expression. So it's like this. Here's a polyphenol molecule, for example. So it has got to get itself absorbed into the intestinal epithelial cell. It's then got to make its way across this intracellular space. It's got to get into the capillary. It's got to travel into an artery, travel around the body, and finally, it's got to get into the cell. If we look at something like the polyphenol epigallocatechin gallate, which is the bioactive in green tea, you see this great big bulky molecule. It is just really too big to go through any of these membranes to any significant extent. And so a lot of the time we talk about molecules like green tea as being antioxidant, and that's partly true because where it's antioxidant is in here in the intestinal lumen. So if we were going to eat a barbecued steak which was covered in blackened carcinogenic oxidant compounds, if we had a cup of green tea with that, which is what the Asians would do, or a glass of red wine if we were more Mediterranean in our inclination, those polyphenols will now quench radical species right here in the intestinal lumen, and that is now going to reduce the burden of oxidants that have to go through the system. So there's certainly benefit in polyphenols, but it's not what we thought. When we talk about them being antioxidants, I don't get here. Less than 1% of most polyphenolic molecules will actually get to the tissue target cells. What will happen to them further down, however, is the microbiota will chop them up into little pieces. And so we will be able to absorb fragments of those molecules. But we don't know enough at this point about what types of microbiota will break this molecule down into what types of smaller fragments. It's a very active area of research and there's lots yet to learn. So then if we look at sulforaphane by comparison, this is why sulforaphane is a standout different type of molecule. It's not a polyphenol, as you can see here. It's just a long, thin, low molecular weight, fat-soluble molecule that has no trouble gliding through these membranes and readily gets into cells. So you get about 80% bioavailability with sulforaphane. Simply means you get about 80% of what you consume in your target cells, whereas you're probably only getting about 1% of these. So the thing to note here is to get a nutrigenomic effect, there's two key principles to remember. There's the dose response, which we mentioned before. If you haven't got a high enough dose, you won't reach the threshold for gene expression. And you have to have a bioactive compound with sufficient bioavailability to reach the target cells. Let's talk a little bit more about sulforaphane and how it's formed, because there isn't any in the broccoli sprouts and there isn't any in the broccoli vegetable. It's actually found in the plant cell as an inactive precursor, glucoraphanin, in one little vesicle within, or vacuole within the cell, and the myrosinase enzyme is in a separate vacuole within the cell. When you chew or bite or chop it, the myrosinase enzyme attacks the glucoraphanin and that produces sulforaphane. What happens if there's no myrosinase enzyme? You get no sulforaphane. Very simple. So when you cook a cruciferous vegetable, the myrosinase enzyme is heat sensitive, so you will destroy the enzyme. And if you steam sterilize broccoli sprouts, which some manufacturers do, to make a sulforaphane supplement, you get no myrosinase enzyme then. And I'll tell you how much steaming and boiling is involved shortly. And if you purchase a product called a broccoli sprout or a broccoli seed extract, there is no myrosinase enzyme there because the myrosinase enzyme is deliberately removed in the manufacturing process. Sometimes they put some back. Traditionally, they haven't put any back. And so what these supplements are, they're just containing the precursor. They don't have any enzyme at all. What I prefer to work with is 100% whole broccoli sprout material that has been able to retain the glucoraphanin and retain the enzyme. And so if that powder, the material is powdered and dried and kept free of moisture, as soon as you add moisture, which would happen if you stirred a powder into a glass of water or a capsule dissolved in your stomach, the myrosinase enzyme will act on the glucoraphanin and you will produce the sulforaphane. So what happens then when you do cook broccoli? There's been quite a lot of studies that have looked at this because it's important that we know. Ideally, if you eat your broccoli vegetable raw, you will retain the enzyme, but unfortunately, the broccoli vegetable contains only about 10% of the bioactives that are in the sprout anyway. So if you microwave your broccoli vegetable for 2 minutes, you've killed all the enzyme. And if you if you steam your vegetable for 7 minutes, you've killed it all too. Now, 7 minutes doesn't actually produce broccoli vegetable that's softened in the manner that most people eat it. So mostly when you cook the vegetable, you've killed the enzyme. When you cook it in, as a rough rule of thumb, you could suggest that you get about 10%. Now, how do you get anything if you kill the enzyme? So if you look at this factor here, so your bioavailability is about 30%, 37%. This is vegetable, not sprouts. You've got about 37% there. And then when you cook it, you've got about 3.4%. So that 10% is your rough rule of thumb. So about 2011, a group of researchers in the US were getting pretty annoyed that there were these broccoli seed extracts and broccoli sprout extracts coming onto the market, selling them as capsules. And what the manufacturers would do, they would refer to all of the extensive database of sulforaphane research and use that data to support their product. The problem was their product didn't have any enzyme in it. So this research group did this particular paper. They even named the product, which I won't name now. You can look it up if you want. But the comment here was essentially that these supplements, people are buying them thinking that they've got a certain sulforaphane yield. They're not getting what they're thinking they're getting. And the real trigger for this research was that you only have to do a little bit of research on sulforaphane to find that it's considered to be highly protective against cancer. And so therefore, it's the most vulnerable people who are buying these supplements, and then they get them home. Well, they don't ever find there's nothing in them because nothing on the label tells you. So they believe they're getting the equivalent doses of the isothiocyanates, which is the sulforaphane family. Here's a graph from the same paper. So this is the blood level of sulforaphane on the left axis here. The green line is the sulforaphane concentration over time. So people took either broccoli sprouts, they took this inert supplement, they took alfalfa sprouts, and they took a placebo. So you can see without a doubt that the broccoli sprout supplement produced high levels of in the plasma, whereas the inert supplement didn't. That little bit that you're getting there in that blue line is the gut microflora, which have limited action in any case. So what they looked at here was that the gut microflora do exhibit some unpredictable but highly variable myrosinase activity. The problem is we don't exactly know which microflora species are having the greatest effect, and we can't quantify that. And the reality is, as you would understand, that a lot of the people who are coming in as patients to your clinic have perturbed gut microflora anyway. A lot of them will have been on medications such as antibiotics that have destroyed whatever they've had. A lot of them will have dysbiosis. So they can't rely on that as a means of obtaining the necessary sulforaphane. So what they did show in this paper is that perhaps about 8% of the isothiocyanates were recovered. and this is a rat study here, but it's the same in humans when they've been given a diet without the enzyme and they're just relying on what's coming from the microflora. It's important, I think, to talk about safety. So this remarkable supplement has had adequate safety studies done on it. There's no evidence of any clinically significant adverse effects that could be attributed to the sprout administration. The only addition to that I would make is that patients with severe dysbiosis will often get pretty uncomfortable gut responses in the short term if they start on a high dose initially, because sulforaphane activates antimicrobial compounds in the gut which will kill the pathogens, and you will get some die-off, and it will often cause a lot of gut discomfort. And all that needs to be done is the dose slowed right down until you find the dose at which the patient is comfortable, and then you gradually build it up to the full dose. So is it actually possible to produce a bioactive sulforaphane yielding supplement from fresh sprouts? And of course it is. And this was another of my review papers from 2013. And in this paper, which unfortunately is not free online, it talks about the extracts or broccoli seed extracts in the way that I've explained to you before. Because what a lot of these products put on their label is, instead of glucoraphanin, the precursor, they write sulforaphane glucosinolate. It's not a scientific term, it's a marketing term. But unfortunately, when you see the word sulforaphane on the label, you go, yes, that's it, that's the one I want. But in fact, these are the supplements which are completely devoid of the myrosinase enzyme. Here's an example of a label like that. So this is an American supplement, sulforaphane glucosinolate from broccoli extract seed. So that's all it tells you. There's no myrosinase enzyme in that. Here's one that doesn't say terribly much at all. In most jurisdictions, especially in Australia where I am, you're not allowed to talk about sulforaphane on the label, and the reason for that is there is no sulforaphane in the product. What's in the product is the glucoraphanin and the myrosinase enzyme, which produces sulforaphane when you consume it. It's very confusing. And really quite concerning that you can't say on the label what you really want to say. There's just 100% broccoli sprout powder, and that retains the myrosinase. This one's really amazing. This is a very well-known American brand, and I'm not going to tell you what it is. But on this label here, it says organic broccoli whole plant powder. So this is the vegetable. This is not the sprout. So there's 100 milligrams of that. I don't know why they bothered. There's 400 milligrams of organic broccoli sprout powder. You see here, they say it contains 140 micrograms of sulforaphane. 140 micrograms, incidentally, is nothing when we're talking in milligram doses. So there's the first problem. Yes, it probably contains some glucoraphanin, it probably contains myrosinase, but micrograms is laughable. So when you analyse it, what you see is here that this 140 micrograms is 0.14 milligrams. So I'm looking for a supplement which has, say, around a 500 milligram supplement, which has or is able to yield at least 10 milligrams sulforaphane. When you do the sums on that, if I want to get a 20 milligram daily dose, so it'd be 2 of these tablets or capsules, I would have to take 143 of these capsules a day to do what 2 of these are doing. That is a 57-fold difference in potency. I mean, this is really criminal. And the other thing that follows on from this, I think, is that people sometimes say to me, Christine, you say this product's so wonderful. How come I've never heard of it? Why don't I know about it? And I would suggest to you the reason you may not know about it is rubbish like this is on the market And if people buy it because they read all the good things about sulforaphane, they'll take it for a month or two, nothing will happen. There's no dose in here that's going to increase gene expression. Absolutely not possible. So keep that in mind. Okay, so let's talk about sulforaphane clinically. There are multiple mechanisms. Mostly I've talked about the NRF2 activation, but that's only one. It's used chemopreventively. It's used in type 2 diabetes, and these are all published studies. And used in liver function. It's remarkable as a liver detoxifier. Also, quite a few studies done on its role in the central nervous system, and we're going to come to autism shortly. So there are all sorts of properties which are secondary to the NRF2 activation, but we're beginning to discover now that there are lots of other properties which are also relevant. One of the most powerful that's worth knowing about is its anti-inflammatory effect. When I was in clinical practice, I think I thought that either omega-3 fatty acids or curcumin were the preferred anti-inflammatories, but I don't think that anymore. And it's reinforced by this fairly recent study, 2018, which looked at healthy overweight subjects and looked in terms of their inflammation markers. So you can see here that we're looking at interleukin-6, and we're looking at how that's tracking over a period of time. So it's quite a long study. So we did 70 days here, went out to 90 days, and then continued on for some time thereafter. And so this one here you'll see with the interleukin-6 at day 70, inflammation was dropping markedly as interleukin-6, and interestingly, over the next 3 weeks, it continued to drop, even though the supplement had actually stopped at this point here, and then it gradually rose. CRP, it dropped, not quite as dramatically, but as soon as the supplement was stopped, it increased. Now, we don't understand exactly how that happened, but it's really important to realise that this is pretty powerful anti-inflammatory effect. And you can do that on a dose of about 20 milligrams a day. What you see here is a list of the selected clinical trials that have been done on a range of different conditions here, from autism to respiratory issues, Helicobacter infection, gut mucosal repair, diabetes, prostate problems, and so on. So in here, you'll see a range of doses, which I just tend to look at in terms of aiming for around about 20 milligrams a day is my starting dose usually, unless someone has severe gut problems, in which case I slow it down. And you'll see here there's some studies have been done with fresh sprouts. Fresh sprouts are not all that easy to get someone to take therapeutically. You need a fairly large quantity daily. They're not all that pleasant, and growing them yourself can sometimes be difficult. It's also very difficult to know what the quality of the seed is, and if you don't have enough glucoraphanin in your starting seed, it'll be very difficult to get the doses. This is a clinical trial here which used 180 milligrams of glucoraphanin without the enzyme, and it had 0 clinical response. So there's your typical doses. The dose for prevention, we don't really know. I think 10 milligrams a day is a good, solid preventive dose. Therapeutically, as you can see here, you may need to take more. There have been quite a lot of papers done on its chemopreventive activity, and this one's actually in a drug development study. As I mentioned before, there's a lot of drug research going on in this space now. The important thing, I think, is that it's safe and it's relatively non-toxic, and it exerts its anti-cancer activities through multiple mechanisms, part of which is involving the detoxification pathways. There's also a whole host of in vitro studies now we won't go through these in detail, but essentially what they're doing is they're combining sulforaphane with one of the chemo drugs and finding that they can increase the dose of the chemo drug because the sulforaphane is protecting the normal cells while the chemo drug is able to do its work. This one here is just illustrating the way in which it dose-dependently upregulates glutathione synthesis. So this is the glutathione levels here, and these are the different concentrations. So these are micromolar concentrations. The 2.5 micromolar concentration here is easy enough to achieve with supplementation, and you get about a doubling there of your glutathione synthesis, which is practical. The 2.5 micromolar, sorry, the 5 micromolar dose here, you'd have trouble getting that in an ordinary day-to-day supplementation regimen. But look, doubling glutathione is a great response. In terms of cancer chemo prevention, and these are in vitro studies, so you can give moderate doses, preventive doses of sulforaphane, and you can prevent initiation and promotion of neoplastic cells. But in order to actually cause suppression, you need therapeutic higher doses. These are only in vitro studies. We don't know whether this is effective or not. I prefer not to get involved in this area, but I'm certainly interested in working over here in the prevention space. I'm just going to skip over this one very quickly, but just to show you that a typical diet of an average person will get about this much quinone reductase activation from the amount of sulforaphane that they consume. But if you're looking at 20 to 40 milligrams daily, which is a couple of capsules, you can get up into this therapeutic range. And I want to talk about quinone reductase now because I think it's the best kept secret in nutritional medicine. It was discovered a long time ago that it's the thing that prevents deleterious effects of redox cycling of quinones, and quinones are the end product of estrogen metabolism for starters. So you don't want that half degraded estrogen, quinone, to attach to your DNA, because if you do, you're going to get a mutagenic response. So quinone reductase is a very powerful preventive enzyme against mutagenesis. Here's our diagram again here that we saw earlier. There's NRF2, just reminding you there's the quinone reductase there. And as I said, if the mutagenic metabolites of DNA are caused because quinone reductase didn't do its job, you're going to start smashing up your DNA and all sorts of things will happen. So quinone reductase blocks that. I hope your Nutrigenomic report shows you quinone reductase because that's one of your most powerful enzymes and genes. So the original research that showed this, breathe a sigh of relief, we're not going through it, but Professor Cavalieri is one of the pioneers of the estrogen detoxification pathways. And essentially, this is the phase 1 detoxification over here, and this is phase 2, which we're going to look at in a moment in a much more simplified fashion. What I want to show you here is in the functional medicine space, there's lots of talk about COMT being part of the phase 2 detox process. Yes, it is, but it is only the very beginning of it. There are far more important steps that continue on out past COMT, and these are the ones we're going to look at here. So if we've got an estradiol molecule here that has to be broken down to a hydroxy form, to a semiquinone, a quinone, and if that quinone gets to your DNA, that's what causes mutagenesis. Here's phase 1, so there's the cytochrome P450 enzymes. Pardon me. So got a 1 site 1B1. If you're in the habit of using DIM as a supplement, DIM actually inhibits this step here. So you're going to reduce the amount of estradiol that's going down this pathway. The next step, however, is, so we've hydroxylated the estrogen. COMT now acts on that, adds a methyl group, and now we produce methoxyestrogens, and those methoxyestrogens are now glucuronidated, and you would, pardon me, sorry, perhaps have a UGT enzyme now which excretes that water-soluble compound. You also have superoxide dismutase acting in here, which blocks this semiquinone. So COMT usually can't take all of the hydroxylated estrogen. What it can't take goes to the semiquinone, superoxide dismutase tries to take care of it. Then it goes to a quinone, and finally to the DNA adapt. How does nature intervene? Well, here we have the glutathione transferase enzymes and its accompanying glutathione. That produces glutathione conjugates, and those conjugates are now eliminated. Now again, the quinone often is still too much to be taken care of by the GST, and we could still cause damage here. What quinone reductase does is it grabs the remaining quinone, drags it back to the beginning, and represents it now to COMT. By that time, COMT has now dumped its load of methylated estrogens as is back ready for more, and now it'll pick up some of those estrogen quinones and take it through the cycle again. So it's the quinone reductase which allows these estrogen metabolites to keep recycling. When you're looking at hormone tests which stop at COMT, you have all this missing information here which just makes such a huge difference. So if you're looking at your quinone reductase genes, NQO1, keep this in mind, because your patients who have got SNPs in the GSTs and SNPs in quinone reductase, NQO1, are the ones who are at greatest risk. So there's your DIM coming in there. Sulforaphane is activated here. Sulforaphane is active here. These are all NRF2 target genes in here, and quinone reductase. And you can see sulforaphane just plays such a huge part in this process. And you can use DIM and sulforaphane together to get your maximum response. So what upregulates quinone reductase other than sulforaphane? This is out of my 2016 paper, and I think it's again in the 2019 one. If you look at quinone reductase activation here, so here's a whole range of different compounds you will be familiar with. There's a green tea we talked about before. Scarcely has any ability to upregulate quinone reductase. Is sulforaphane over here, which is by far the highest. Andrographis, not too bad. Quercetin, very little. Silymarin, now we look at silymarin as a major liver detoxifier, but its effect is minimal. DIM has a very tiny effect here on quinone reductase, but its main role is in phase 1. So other applications, the other thing that's really important for sulforaphane is its ability to eradicate Helicobacter pylori, and there have been 2 good clinical trials done on this. And it's largely because the Helicobacter organism produces an enzyme called urease, and it's urease which breaks down urea that's coming from proteins in the diet. So when urease breaks down urea, it produces ammonia, and ammonia increases the pH in the stomach. So the stomach, which is largely acidic, now becomes alkaline. And that's exactly what Helicobacter likes. It likes an alkaline environment. So it's carved out a nice little niche for itself where it takes up residence. And in the process of doing that, reducing that pH, or increasing the pH level, I'm sorry, you're going to reduce mineral absorption. and you're going to produce gastritis because now we've got a lot of erosion in here, and protein digestion is going to be inhibited because we have this impairment in pH. So how does sulforaphane get into the act? Sulforaphane, through an entirely different mechanism that's got nothing to do with NRF2, is a urease inhibitor. And so that is just such a clever little mechanism, which I think has very, very valuable applications. We see such a lot of patients taking proton pump inhibitors and so on, really causing themselves all sorts of grief. That's a story for another day. But here's a very simple way of dealing with that, and that dosage will be on the notes when we send you the PDF of the slides. You'll see the dosage that is used for the Helicobacter trials. In neurodegenerative disease, a lot of neurodegenerative diseases are related to oxidative stress, and conditions like Alzheimer's and Parkinson's disease may not be totally due to an oxidative environment, but it's certainly a huge component. So oxidative stress, inflammation, and neuronal loss all accompany these diseases. There are no good drugs available to deal with any of these, but this paper here concluded that sulforaphane appears to be a promising compound with neuroprotective properties that may play an important role in preventing neurodegeneration. And this paper here is talking about enhancing cellular defenses and a guardian of redox homeostasis. So this is talking about brain inflammation again, and the way in which the microglia in the brain are continuously producing free radicals and inflammatory cytokines. And here we show that we can use NRF2 activators in order to reverse some of those effects. Getting almost to the end. So in terms of autism, there have been 2 published papers on autism, and this one came out at the end of 2014, and it was met with great excitement for pretty obvious reasons. And it was a clinical trial, and what they've done is they've taken 40 participants, 26 in the active group and 14 in the placebo group, and then they've done an 18-week run out where the participants didn't take anything. So what they looked at here, so this is a graph looking at aberrant behaviour and the change in scores. So the red line is the placebo group and the blue line is the sulforaphane group. So keep in mind that the sulforaphane was given for 18 weeks. So you can see the behaviour gradually improved and finally got to this point. But you can see when the sulforaphane was stopped, the behaviour tended to relapse. Now keep in mind, this is just a single intervention study. There was no other therapy involved. And I think it's pretty remarkable that you can do that with just sulforaphane alone. This graph here, same study, now they're looking at social responsive. So this is eye contact and communication and so on. Similar pattern, improved to 18 weeks and then tended to relapse. But we talk to lots of clinicians in various countries around the world who use sulforaphane now based on this study and our recommendations for the doses that will do the job. So in general, we think of sulforaphane as having a pretty bright future. There is a continuing increasing number of studies being published to show its benefit. Some of the studies are just in vitro studies, but there are enough clinical trials to give us the confidence to know we're dealing with a pretty special molecule here. And what the conclusion here of this abstract is that says that it's in normal cells, it has protective actions, and it could be considered as a phytochemical at the forefront of natural medicine. And with that, and Catherine, I'll hand back to you.
Excellent, Christine. And the questions have just been popping in on the side. some practical things from like, when is it best to take it? Morning, evening, empty stomach?
The empty stomach is really just dependent on the patient's tolerance to taking anything on an empty stomach. It doesn't make a great deal of difference. Anything that you're taking for its nutrigenomic activation will switch on those genes, and then those genes and the products of those genes, the enzymes, will last for several days. So it makes no particular difference when you take it.
Okay, and it doesn't impact sleep as well, or if you take it in the evening?
Some people will say that when they first start taking it, it'll keep them awake. I don't know how many people, some do. I mean, take it in the morning, probably easiest.
Super. Then questions in regards to the goitrogenic effect in regards to Hashimoto's and thyroid.
So the good thing about broccoli sprouts is they don't contain any progoitrans, and therefore when they're acted on by myrosinase, there's no goitrogenic effect at all. The progoitrans develop as the plant matures. So as the tiny sprout grows into a vegetable, that's when the progoitrans develop. So that's good news. And also doesn't contain any vitamin K1 in the sprout, so it doesn't affect a patient on warfarin.
And then if, because now we are into the pharmaceutical drugs, then any interaction with proton pump inhibiting drugs?
Look, nobody's ever done those interactive studies, so we don't really know. In theory, because sulforaphane activates NRF2 and all of the detoxification enzymes, theoretically, anything that's upregulating phase 2 detox could reduce the efficacy of a drug by degrading it somewhat. More than that, I can't say. But then so too, what if you're eating your 600 grams of vegetables a day and living a nice lifestyle, you would also detoxify those drugs. Because really what we're doing with this, we're mimicking what a healthy diet should really do in terms of its plant content and exercise, sunshine, fresh air, etc.
And well, with a lot of functional medicine treatments, people will be able to get off the PPIs anyway. So that's
well, you'll get off it with this too. Yeah, we use sulforaphane. I mean, a lot of people taking PPIs have got a Helicobacter infection anyway, and they have severe dysbiosis, not just down in the intestine, but right through the system. Sulforaphane does a wonderful job of dealing with those issues.
Another question came up in regards to estrogen, and that was like, well, I have for sure experienced a patient getting hot flushes on Roculux, and then you're not that popular when you give a patient hot flushes. But I guess it's because you're just detoxifying the estrogen faster, and then you get these
yeah, whenever you get any adverse effects, I just slow the dose down and allow the system to catch up. Some people say to me, there seems to be a misconception, we need to talk about this. People think that sulforaphane is reducing their estrogen. It's not reducing it. It's just sending it down the right pathway, and the things we talked about with quinone reductase, where it's safely detoxifying it, that is not reducing the level. If the patient's getting hot flushes while they're taking it, I would suggest just slow it down and just keep working with a variety of other factors until it all levels out. You're aiming for homeostasis, and clearly you haven't got it at that point.
Okay. And what in regards to patients on tamoxifen?
Nobody knows. Again, we don't know what the interactions are. I mean, these are tricky questions. It's like patients on chemo. I just suggest people stay right away from dosing when a patient's on chemo. I think just let the oncologist do his thing. As for tamoxifen, it does have a mild NRF2 activating effect, among other things. Whether this has any adverse effects taking them simultaneously, we don't know. No one's ever done the study, so I can't answer you. I'm sorry. I can't give you the answer you want.
That's okay. That's fair enough. So when you had this beautiful slide up in regards to the therapeutic dose, but what if we take Roculox as a what is, because that's 700 milligrams.
Okay.
Active,
So so a 700 milligram capsule has got about 12 milligrams sulforaphane yield.
Super, because then we can use that chart as a very clinical.
Yes, well, and I do that. I do that too, because ours are also 700 milligrams here in Australia. And we just say the standard dose is 2 caps a day. Most of the time we're aiming for 20 milligrams sulforaphane yield a day. That's our standard starting point, and we go up or down depending.
Yeah, super. Because then we can, when we get the slides, we can take a screenshot and print it out and have it on our desk.
Yes,
well,
you soon get used to it though. You'll know what it is, but yes, you can cheat for a little while.
Of course. So anyway, Christine, the time is up. People have to go and enjoy their evening or run off to work and so on. Thank you for all the questions coming on the side, and a lot of them were actually answered while you were going through it.
I guessed a few on the way through, which is why I ran rather late, because I was chatting a bit along the way. But anyway,
no stress.
I hope it's been useful.
It certainly has. And of course, as usual, we will send out the recordings, hopefully tomorrow, if IT allows. And Christine has promised to send me the slides, so we will share those too. And of course, we will send out invites for the next webinars. I think it's going to be on some gut health lab stool testing, something like that. But we will send out invites. So thank you, everybody. Christine, it's been amazing, fantastic. It's been a great learning curve, I think. And if nothing else, for those out there who already knew a lot about this, it has been comforting to support the knowledge that you already had. So it always works in 2 ways.
Yes, thanks so much, Anne-Catherine, and thanks everybody for being part of the session tonight, or today for you.
Thank you. Take care, everyone. Virtual hug, and see you soon, hopefully. Take care. Bye.